Good evening, everyone. I would like to welcome you all to our second talk of our series, Understanding the Management of Important Zoonotic Diseases in Africa. And a warm welcome to our students from TUT, who are our primary audience today. And today's topic is research and surveillance of zoonotic diseases with an example of a case study from Congo. And today we have a very great speaker. She is someone who started out as a public health immunologist and started with her career in vaccine research. And then she started veterinary and now is working on One Health projects, focusing on investigating and assessing the spillover risk of viruses such as Ebola in human-wildlife interface, with particular focus on bats and eggs. And also, she's looking into their conservation. And this person is Dr. Eeva. Dr. Eeva, we are so happy to have you as our speaker today, and please welcome.
Thanks very much, and thanks Stella for the introduction, and thanks for the whole Share Screen team for inviting me to talk, and it's very nice to talk to you, you students. Very much looking forward to give you a bit of an example of what we're doing in Congo. I work for an organization called Wildlife Conservation Society, and this work that I'll be presenting to you today is done mostly together with them. It's a very big organization. We've got projects in 56 countries globally at the moment. So a big organization, big conservation organization, big employer. That's my advertising talk. Right. Yes. Yeah. Do if you are struggling with a context or an explanation that I'm giving, you can always put your hand up. I'm happy to explain more. It's good that we take an opportunity to make sure you understand kind of what I'm talking. Right. Yeah, really to begin with, Stella asked me to give a little bit of my personal story. She already explained a little bit, but also just to give you a little bit of a story of how did I end up doing what I'm doing now. And it's been a kind of journey. Like Stella mentioned, I started as a research scientist in public health. So I was working in a laboratory mostly in United Kingdom, in a public health research institute. And during that time, I got training to dealing with dangerous pathogens. We were doing vaccine research into things like anthrax and smallpox at the time, vaccines that are now used for Mpox epidemic that is happening. And because of that training, I got then sort of back in 2014, I got sent to West Africa. There was a big Ebola epidemic happening in several countries in West Africa in 2014 to 2016, and with this training that I had of handling and doing diagnostics with these dangerous pathogens, I got sent over to run these field laboratories in Guinea and Sierra Leone mainly. And I spent several months there, and I really enjoyed this work. I felt like I really had found my thing, and I enjoyed being in a field and kept going back. And really during this time, I then decided that I will not go back to the research laboratory in the UK. And I had always been a conservationist really at heart since I was very small. That was my sort of personal interest. And I thought, okay, this is my opportunity also to jump completely from one area to another and go into conservation. And I was very lucky. I found a great ape conservation project in Central African Republic. And they were looking for someone to run diagnostics and their field laboratory in Central Africa for same diseases that I had been looking at in humans. So Ebola's, a lot of respiratory diseases, things that we get infected as well. So zoonotic diseases. So I could offer this skill to them, so I was in, and I moved to Central Africa. And during this time, I worked a lot with the vets that were in the field. And I always found them very inspiring people. I respected their work a lot and found it very interesting. I was assisting in snare removals. We're taking a snare here on the bottom left corner picture. We're taking a snare from a female gorilla. I was doing a lot of necropsies with the vets, as well as then running the lab, the field laboratory. But it was these little guys that really made me finally become a vet. So just as I was joining the project in Central Africa, there was a big problem with poaching of African grey parrots. So they were caught in the wild and sold and exported to pets, mostly outside Africa. And really, kind of in these first few months that I was there, we received so many of these. We landed with over 1,000 of these parrots in our hands in a space of just a couple of months. And their flight feathers had been cut so that they can't escape. They were stressed, they were sick, and just generally in a really bad shape. And despite our best efforts, they just kept dying, and we couldn't release them because they couldn't fly. And this is where I kind of really realized how valuable these hands-on skills, practical hands-on skills are really. And that decision, this is one of the pictures of one of those confiscations that we had. And that really kind of made me decide that I'm going to be enjoying this work in the field in the future, so I'm going to become a vet. And so I did. So I joined a vet school in London, and during this time I carried on working in Congo in these projects that I was already involved. So that is now closer to 8 years that I've been involved with the project in Congo. So that's my story. A good example is never too late to follow your dreams and your ideas. And really now then, with the rest of this time, I would like to just give you an overview of 2 different projects that we have in Congo that I've been involved with. They're both kind of combining both conservation as well as this kind of public health element in them. They are surveillance projects. One of them is a research study, and the other one is actually an active sort of ongoing surveillance project. And both involving zoonotic diseases in Congo. So just to make sure that everybody's kind of on board with what we're exactly talking about, and I'm sure you are. I think I was told that you've been doing some of this theory already. But just to recap, if you've heard of a One Health concept. So One Health is this kind of idea and an integrated and unifying approach that aims to balance the health of humans and animals and ecosystems. So it's really kind of this understanding and concept that human, animal, and environmental health, they are all interlinked. And then we need to kind of work towards solutions where all these elements are considered when it comes to health, conservation, everything that we do in this space. So conservation is really integral to the objectives and the way we function in this One Health idea. So One Health is about healthy environment and healthy ecosystems, and it's about healthy and stable wildlife populations as well. So conservation is really important part of this. And then when it comes to diseases and infectious diseases, we are all linked because we share a lot of the same pathogens. So zoonoses that infect both humans, but also then animals and wildlife and domestic animals. So this is kind of the focus that I want you, or sort of like the concept that I would like you to keep in mind as you listen to this. Right, so the first project that I would like to introduce you to is a community-based, what we call a community-based wildlife disease surveillance for detecting Ebola virus epidemics. So epizootics is a word same as epidemics, but in the animal population. So it doesn't involve humans at that point, but then it's also an early warning system for these Ebola outbreaks that might be spreading in human population as well. So this is kind of a really nice example of a project that has both conservation as well as public health objective in the heart of this surveillance. And just to kind of get you on board with this idea, I just need to give you a bit of background, a bit of history. So here's a map of Central Africa, and all these little purple dots are past outbreaks of Ebola virus disease in humans. So back in the sort of mid-2000s, in the Congo Basin especially, there was a big cluster of these cases going on, or these little small epidemics in the rural communities. Since that, we've had big ones, the Western African one that I mentioned earlier. There's been several in recent years in the Democratic Republic of Congo as well. So they've been varying in sizes, but what we know from these, especially these kind of smaller epidemics that happen in the very rural parts in the deep in the forest, is that they started when local hunter-gatherers were finding dead animals in the forest, and they would collect them to eat. So a lot of the start of these little epidemics started like that. They were traced back to dead animal found in the forest. And these were really happening, like I said, sort of in the 2000s and mid-2000s. And as well as human epidemics and human deaths, these epidemics affected really badly great apes. So at the same time, there were areas where almost complete populations of gorillas and chimpanzees were wiped out in just a matter of weeks due to Ebola. So they were also infected very badly by these outbreaks happening in the forest. And this was really so severe for the great apes that suddenly transmissible diseases, and especially Ebola, was raised as one of the top threats to the existence of great apes in this area in Central Africa, together there with poaching and habitat loss. So this kind of really raised at the time the importance of transmissible diseases in this kind of wildlife conservation area. And what we now know, for example, you might be aware there's for the last couple of 2-3 years already now, there's been an avian influenza going around whole world, and that is another example where it is so severe in places that it is actually threatening the existence of whole species. So really what we then were faced with when these animal deaths as well as human epidemics were happening in the forest is we kind of had a need to try to understand where do these Ebola outbreaks are happening. But in the context of Congo, it's really quite difficult. So this is the kind of environment that we're dealing with. It's a very dense growth tropical rainforest. Going around there doing surveillance and trying to look for carcasses is very difficult. It's hard to get into the forest. So what we then came up with to gain information about these potential wildlife deaths and mortalities that were happening in the field, we decided to contact the people who are actually already there. So this is the hunters and the small rural communities that are living in the forest. So what our vet team did, they started to go around in a car around all these little villages in northern Congo. And they would explain to the communities, to the hunters, the kind of risks that are associated with eating dead animals. So they might be infected with disease that can also infect humans, or zoonoses. So they would run these kind of educational programs, explaining the diseases and the viruses. How do you get infected and how can you protect yourself from being infected if there are if there are Ebola carcasses out in the forest. And at the same time as we were doing this kind of educational program, we also asked the hunters to actually let us know if they can find if they find some dead animals in the forest. And they did. And when we would get one of these reports of dead animals and carcasses, we'd quickly dispatch a team to go take a sample of these carcasses. So we are dealing with carcasses that have potentially something very dangerous like Ebola in them. So we have to be very careful. How do we take the samples? We have very strict protocols of how to put our protective clothing on, how to take the sample, how to take the protective clothing off to avoid being infected ourselves should there be a virus present in that carcass. So we take a sample, then we need to test it to see whether there is Ebola virus or not. And sometimes we can do this in the field. We've got some equipment that allows us to actually do the diagnostic test right there next to the carcass immediately, or sometimes we transport it to the laboratory. And then as we get a result, we share that result with the community. Since the start of this surveillance program, very luckily, we haven't had any outbreaks of Ebola happening in the forest. But nevertheless, we do go back to the communities, share the information and the results, encourage them to keep reporting carcasses to us. And then we just do a little recap of that educational program about the risk of zoonoses. And it's really kind of building this kind of rapport and understanding over several years now with these communities. And even though we haven't actually detected yet active Ebola outbreak in the forest, this kind of program really does potentially still have an effect. So it has an opportunity and a potential for preventing other human disease outbreaks as well through this behavioral change. So we're really encouraging people to avoid eating carcasses because they are potentially dangerous. It's not just Ebola, that's kind of mainly what we're testing for, but there are other diseases also that can be transmitted the very same way. And then at the same time, we always keep thinking of strategies. How can we protect great apes should this kind of big outbreak happen again in the forest and within the great ape population? Luckily, the population of great apes and gorillas has bounced back since that, so it has recovered somewhat. Not quite to the same level that it was, but it has. So it's not quite so critical, the situation right now. And here's a map, just to kind of explain. All these little green dots are villages that we visited. They're usually the villages are kind of around routes, so that's why they're kind of in the strings like this. We visited way over 300 villages by now as part of this this work, and reached about 15,000 hunters and women and children who are living and hunting in these areas. So this is really kind of covering potentially quite a big area, and definitely an area that we could never ever cover with any other kind of active surveillance method. There's no way we could be looking for dead animals ourselves within our conservation organization team, vet team, like this. So this is really about making use and working together with people in this kind of wholesome project that helps both us doing the surveillance, but should also be helping the communities and should also be helping the wildlife in terms of preventing or at least spotting very quickly a disease. So this kind of, we call this a sort of early warning system, if you like. And like I said, this surveillance program is still going on. Our vet team car is still going around villages and reinforcing this message. So we really carry on this all the time. Right, then this brings me on to the next example that I wanted to give you. This is kind of something. This is a project that we started in 2022 initially. And it really started, we when our team started to notice that there's an increasing number of bats being sold in the markets of Brazzaville. So they were sold for food consumption. And we really started to sort of notice an increase of this and started to look more into it. And what started as kind of a research study to start to initially understand what is going on, this is also now molding into more of a surveillance study. So we are going to be continuing this in the long term in some form. What I'm presenting you today is just kind of giving you a little bit of idea of the methodology of what we've done here and kind of the early findings. So this work has not quite finished yet, even around this initial study, but I'll just give you a bit of an idea of what's going on. And just like the previous case, this is also kind of a really nice project combining both conservation and human health concept. So bats are consumed in many parts of Africa and in the Republic of Congo. And like I said, we think that this trade and this consumption is increasing in Congo. So the bats, they provide a very important income source for those people who do hunt them and trade in them. And they're also an important protein source for the people who do eat them. Not everybody eats them, but there are people who do regularly consume bats. By far the most widely hunted and by far most traded bat is this straw-colored fruit bat, Eidolon. Eidolon is a seasonal migratory bat. It ranges almost all areas of sub-Saharan Africa, all the way to South Africa and Malawi and where most of you are. It's a seasonal. It migrates after fruiting seasons, so it keeps kind of coming and going. These are incredibly important species for the healthy planet, basically. They are seed dispersers across this whole range that they migrate through. They pollinate trees, especially kind of nocturnally flowering trees, and they contribute to nutrient recycling as they move around. So they're very, very important species. Their migrations count to one of the largest mammalian migrations on this planet. So their collective effect is really huge, and they are really kind of, I call them the gardeners of Africa. So incredibly important species. Their numbers have been declining. They are very widely hunted, and their numbers have been declining, and the IUCN now does class them as near threatened, and that their numbers are declining. So they're very important ecologically, but they have also been found to be hosts for many viruses that are dangerous to humans as well. So many zoonotic viruses. We have evidence from studies where we look at antibodies as well as where we look at actual viruses in these bats that they have been infected and they're capable of carrying a lot of viruses, such as these Henipa viruses. I've listed some of them, Ebola viruses, and importantly coronaviruses. So these bats are a natural host reservoir species for coronaviruses, many of them. And then just a little bit about this bat trade. So what's kind of the problem here? So we now know that bats harbor a lot of viruses, and it's this bat hunting and the trade that brings the bats very close to humans. And this close interface creates opportunities for viruses, those pathogens in the bats, to spill over, so jump into human population. So I just want you to have a look at this picture a bit. This is kind of like a typical scene from a bat seller in a market, at a market in Brazzaville, the capital of Congo. So the seller is handling bat bats bare hands, so without any gloves or anything. There's both live and dead bats that he's selling. Sometimes some of these bats stay with him for quite a long time. And so this is a really, really close interface. It's not just like bats hanging up in a tree above your house. This is really kind of a very close interface where the person selling, person buying, and the person hunting is handling these bats. So this is kind of like a really close interface, which we would class as a risky interface as far as the contact, extent of the contact goes. So with this kind of context that we have here, we wanted to kind of understand what is actually the risk. So we can see what's happening, but what do we find in these bats? What is the risk here? What kind of diseases are we potentially talking about? So we really wanted to understand more of the level of risk involved in this trade. So we set up what we call a cross-sectional study to look into the range and the diversity of the viruses that we can find in these bats, just to kind of begin to understand the zoonotic risks associated with this trade. And we took two different towns in Congo. Brazzaville down south is the capital city, and Wesso in the north, which is kind of provincial capital. Both are quite vibrant, and there's definitely a vibrant bat hunting and market trade going on. And what's in our toolbox? So when we look at viruses and start to understand of exposure and viruses that are present in a certain population, what's in our toolbox? We've got basically molecular and serological methods in our hands. Molecular methods, the difference is that the molecular methods, they are looking at current infections in the bats or humans. So that we would actually be looking and detecting the virus itself. And we do this by looking at the viral genetic material. And in viruses, they are either RNA, ribonucleic acid, or DNA, same as with us. We've got DNA as well. So we look at those, we try and detect those that are specific to different viruses through methods such as PCR, polymerase chain reaction, you might have heard of that, and sequencing methods, which is kind of like a more detailed and a bit more advanced method. But these are kind of our usual things that we do. Or then alternatively, and this is often a very powerful way to start understanding initially what's going on. So serological methods. Serology, the word means something to do with antibodies. And we're looking at past viral infections. So every time we're infected with a virus, our immune system reacts to that virus. And that stays for a long period of time. So it kind of leaves this kind of like a memory or footprint, if you like, in the body, both bats as well as humans. And by detecting this, we can get kind of good idea of what historical infections have the bats or the humans had. So we do this, so this is antibody detection basically, and we use different kind of assays for this. One you might have heard of is ELISA, that's commonly used. So that's just to give you kind of like a basic idea. And we went ahead and basically used both of these approaches. So what we did, we went to talk to the hunters. We teamed up with the hunters and asked, can we take samples from your bats? And they agreed. So we sampled them all in all, 453 of these straw-colored fruit bats. We took some swab samples from their nose and mouth, and some poo samples, fecal samples, and then also some blood. And we also asked if we could take a blood sample from the hunters and the market stall sellers who are trading these bats, and they also agreed. So we managed to get 114 blood samples from the people who are working here. And then as any research study, ideally we would always have a human control or control group. So we also took blood from 100 people who don't consume bats, don't handle bats, have nothing to do with bats as far as they know themselves. So that was our sample collection. So then we started to analyze these samples to have a look what we can find. So I mentioned earlier the molecular detection of the viral genetic material. So we did exactly this. So we started to look at kind of wider approach. We looked at families of viruses, and we chose 3 different families. So kind of groups of viruses that are related: coronaviruses, filoviruses, which include things like Ebola, and paramyxoviruses. These include things like Henipa and Nipah virus. So we used this PCR method to look at the RNA, the genetic material, and we found a lot of coronaviruses in these bats. So 52% of all the bats, so over half of the bats that we sampled, had coronavirus infection happening at the time. And 5 of the bats, a lot less common, had a paramyxovirus infection happening. And this we found quite interesting. So in Brazzaville, in the capital city, there was a lot more of these bats were infected with coronavirus. So 63% in Brazzaville, and then in that northern other town, Wesso, only 38%. So there was a big difference in there. We then wanted to sort of have a bit deeper look into these coronaviruses. Coronaviruses are very important subject for surveillance because almost all mammals have got some kind of coronavirus specific to them. And these coronaviruses are also very good at mutating. They're quite fast mutating, and because every single species, mammalian species, seem to have their own coronaviruses, they can also mutate into a new host quite easily. So they are quite sort of COVID-19 is a good example of this. It's a coronavirus that jumped from another species into humans. So this is why it's an important target for us. So then we use this kind of what we call sequencing method just to have a look at what kind of coronaviruses do we exactly have. And each of these lines here on this slide is a sample from a single bat. And this is what we call a phylogenetic tree. So it kind of shows us what the relationship and how closely related these different coronaviruses are that we found from the bats. And we found quite a lot of diversity, actually. So there's probably quite a lot of different, there's a lot of different kind of coronaviruses. Some bats might be even carrying several coronaviruses at the same time. And what we then did is we started looking at if we could relate these coronaviruses that we found to known coronaviruses. And the closest that is known is this a coronavirus that is being detected in Cameroon called Eidolon bat beta coronavirus. So that gave us a bit of information. We have something called beta coronavirus. And what is that? So these are different types of coronaviruses that have been found. This is quite a simple sort of diagram. There's a lot more of coronaviruses, but just to give you an idea that there's alpha, beta, gamma, and delta coronaviruses. And we found this beta coronavirus, which is kind of more closely related to things like SARS-CoV, which is the COVID-19 causative virus. There's MERS, as well as other bat coronaviruses, mouse coronaviruses, and human coronaviruses. So that kind of gives us an idea how closely related it is to some coronaviruses that do infect humans. Right. So that was kind of, that kind of concludes the kind of our molecular investigation into what kind of viruses we can find. Then I mentioned that we can look into the past infections by looking at antibodies that we can find in the blood. We did this kind of work for both the human and the bat blood samples that we had collected. And we have this method that allows us to look at several different viral antibodies at the same time. They've all listed in there, a lot of names, but basically 2 groups. They're filoviruses that include, again, there's Ebola virus that everybody knows about. and Thai virus. Maybe you've heard some of these, but yeah. And paramyxoviruses, Nipah and Hendra are the kind of main ones that we are often concerned with when we talk about zoonoses and things that can infect us humans. So we tested all those bat blood samples and the human blood samples from the hunters and the market sellers to see what kind of antibodies we can find. And we found quite a lot. So this is kind of like this is something called radar plot. So on the left-hand side is all those filoviruses, and on the right-hand side are all those paramyxoviruses. And all this data is kind of the compiled data from the bats, just bats. So there's a percentage, which means that how many, basically, what kind of percentage prevalence of the antibodies we found in this group of over 400 bats. And kind of most important here and interesting ones are on the top left, on the left-hand side plot are Ebola virus and Bundibugyo virus, BDBV. And again, on the right-hand side in the paramyxoviruses, we find Nipah virus and Henipah virus. And then there's something called Ghana virus, which is a bat virus there on the kind of 7 o'clock, if you like, if you look at it as a clock face of that right-hand side graph. So that was in the bats. And then when we looked at the human sera, we found something similar, not quite as strong, but something, and not quite as prevalent, but kind of in a similar lines. Filoviruses on the left and paramyxoviruses on the right. But I'm going to show you a graph that might just highlight a little bit easier the main point that I would like you to kind of understand from here. So like I said earlier, these are still quite early results, so we're still looking into this, but this is kind of like one of the messages that we are now seeing here. So on the left-hand side are some samples from the bats, and on the right-hand side graph are samples from humans. And we can see antibodies in both bats and in humans against this Ebola virus here and the Bundibugyo, which is the BDBV there, the first left side viruses on both of these graphs. So we're seeing antibodies in both humans and bats, and that kind of indicates that they have both been exposed to these kind of viruses in the past. And that also suggests to us that there is potentially this infection going on between the bats and the humans. So the bats are infecting humans. What we haven't done yet, I mentioned to you earlier that we also had a control group of humans who have never been in contact with bats yet. And unfortunately, we have not analyzed that data set yet. We're doing that this month, so we're kind of working on it, but we don't have that data to share with you. And that would be very important to understand strongly whether this is actually, can we draw some epidemiological conclusions from this? So that is kind of a data that is still missing. But what we're already seeing that the people as well as the bats have got the same antibodies. Same thing with these paramyxoviruses. We can see Mojang virus both in humans and bats. It's the MOJV there towards the right-hand side on both graphs. So we see antibodies to those in both humans and bats. But then Nipah virus, in this group of people, we didn't see, and also Ghana virus. We see antibodies in bats, but not in humans. And that is a bat virus, so it is possible that humans just simply don't get infected with that one. But again, there are some indications that transfer of viruses might be happening from the bats to humans. But like I said, this is still early data. We need that controlled group to understand if there's a difference between those people who are actually handling bats and in that risk interface, or and the people who have nothing to do with bats. So that's crucial information that we need. And that is something you always need to make good science. Right, so that really concludes my results of this study. This is still ongoing. But But what I would just like to sort of, again, finish off, we're all kind of conservationists, certainly hope you are all as well. And this is something that I just like to do. So kind of reflect at the end of my talk of what is all of our role in conservation. We were earlier talking with vets, and we were kind of reflecting on the role of vets in this, but we all have a role in here. And one thing that I just kind of thought about is that we are all in different walks of life. We all do slightly different work. We are students. We are already professionals doing something, but we all can share the message and what we know about conservation. So kind of educate people around us with the knowledge that we have. It doesn't matter what it is. We all have something to share. And it's kind of just, I guess, my encouragement to try and say that we all have something important we can say to promote conservation in whatever walk of life we are, in whatever situation in that path. So yeah, that was kind of my concluding remark here. And thank you very much. Here's just a list of all our partners that have been very closely working with us. And like always, these kind of efforts are are a result of multiple partners working together. We can never do these things alone, and we can all contribute something.
Thank you so much, Eva. This was very, very interesting. And I'm just curious. In your presentation, you said in the capital city, there were like high percentage of people with the coronavirus. So I'm just wondering, is it that because in the city, maybe they're eating a lot of birds, or they're getting into contact a lot with the animals, or what's happening? Like, what was the explanation behind that?
So you mean for the coronavirus, high prevalence of coronavirus is in the bats in Brazzaville? Is that what you're talking about?
Was it in human or in birds?
It was in bats.
Ah, I
Yes.
thought it was
Yeah.
in human. Okay.
Right. Sorry. Yes. It was in bats. We didn't test coronaviruses in the humans in this case, and especially with the antibodies, because almost everybody has them now. Almost everybody has been in contact with COVID at least. So we didn't look at this point for the antibodies in humans, nor the virus in humans.
Thank you for that. And I think it will be very interesting for us to see, like, to hear about the results that you will find in the control population of people that have never been in, like, have never been in contact with the bats. Yeah, I'm looking
Yes.
forward to that.
So am I.
Yeah.
Yes,
Yeah. And
I agree.
now I see Karen has his video on. I think he wants to like have a question or comment. You can go ahead, Karen. And for the students, please raise your virtual hand if you have a question.
Absolutely. Yeah, Eva, thank you so much. First of all, that talk was fascinating. It really it does justice to show how we should be bringing together conservation, human health, wildlife health, all under that One Health banner. So I think you really did that justice, and your work it's just fascinating. So thank you for that, and thank you for sharing that with us. I wanted to find out when it came to Ebola. I noticed you mentioned that a lot of the cases in humans in Congo were coming from animals that were already dead in the forest that they had found and harvested. Am I right in that? Now,
Yep.
Was it the case that it was majority of the cases were coming from already dead animals, or were there also cases coming from bushmeat that was harvested through poaching?
So the cases that have been able to be linked were dead animals. So dead animals that were found, and that the individuals who found the dead animal themselves had eaten. I think they were all dead. In this case, I think there was indications also that sometimes wild animals could be weakened by the disease. They are not quite dead yet, but they are maybe about to go that way. So they might be easier to catch. So kind of signs of sickness are sometimes even in live animals just as risky, or something to pay attention to. What comes to sort of bushmeat, are you thinking of something that is in the markets, bushmeat at the markets, or is that what we're thinking?
Currently we're facing in southern Africa. In fact, throughout Africa, we're facing this very large snaring crisis, and often whenever we've spoken about snaring, and snaring is synonymous with bushmeat, and one of the warnings we've always been given is, if we talk about Ebola and how snaring and bushmeat is associated with Ebola, but hearing you say that a lot of the infections were actually coming from animals that were already dead and then harvested in the bush. But I guess at the same time, it's quite easy to kind of mix in dead animals with the animals that you've caught with your snares. So it's hard to see the difference, but it would be interesting to see how many infections, or what's the ratio between a harvested dead animal and carrying that disease and infecting someone, as opposed to an animal that was caught in a snare, which could have been ill at the time or contracted the virus. But I guess that's also a very difficult thing to kind of tease out between that.
Absolutely. And the kind of range of diseases and viruses, or bacteria equally, that can be dangerous to humans, it's a huge range of things that we could have. Some of them are more severe, some less severe. Some of them could be something that, when we talk about something like Ebola, to most mammals that we are talking about here, it's so deadly that and it kind of completely takes over. We've done some laboratory controlled tests of how long does something like Ebola virus persist in a carcass, and it's a long time. It is a long time. For detection, for actually being infectious, it's not that long. So viruses, by definition, they need a live host in order to live. So that's why the kind of the survival of the virus, certainly in Ebola's case, is less. So we're talking about maybe a few days, whereas we can still detect it for diagnostic purposes for several weeks potentially. Yeah. So yeah, it's an interesting one, and we've also kind of started to look into anthrax in these carcasses, and that is kind of like a different thing again. So not necessarily quite as deadly in animals immediately. Might weaken an animal, you might catch that earlier. You could catch that in a snare. So again, that risk is there.
Thank you for that.
Thank you so much, Ken, for the question, and thank you so much, Dr. Eeva, for the good answer. And I see there is a hand up from Isode Virg. I'm so sorry, this name. I'm trying to pronounce the name. Yeah, yes. Would you please unmute and ask your question? Oh, I'm so sorry.
No, don't worry. Isolde is the name. Thank you, Eeva. Very interesting. I'd like to know it's very surprising that all 100 people were willing to give blood samples and participate, which leads one to believe that they are oblivious of, that they're doing something wrong, or, they don't think that they're doing something wrong. How do you see educating them? Do you go from the danger of the virus or the protection of the bats? Conservation-wise, like what approach do you actually take when you interact with them, and were they completely like friendly with you and like not at all looking guilty or feeling guilty or anything?
Yeah, very good question, very important question, and a very important thing to consider when we do these things, because obviously at this point we are still evaluating this, right? We kind of,
yes.
It's obvious that we've got a bit of a kind of can't help but to have a bit of a sort of we've got an idea in our head that this is likely to be dangerous and risky behavior. we can see that there's a conservation concern starting to rise with the increasing capture of these bats, and all of this is kind of there in the background. Now, I mean, I have to say we've got a massive strength in within our organization and in our team, that we've got a socioeconomic team who are professionals in dealing with this kind of thing, dealing with people, explaining situations. And they actually conducted a study, study into this bat trade in a sort of socioeconomic context already before, and made contact with a lot of these hunters and market sellers, and they were very open to share their knowledge. It was quite interesting. I wonder if I can cite how accurately I can cite the numbers, but basically there was quite a big differences in the different groups in that market chain, whether they were aware of zoonotic diseases. So most of the hunters had heard about zoonotic diseases. They had heard about Ebola. They had heard that there's a potential link with bats, and they were pretty nonplussed about it. So I think the situation we have there is that they see that they've been doing this for a long time. They haven't got sick. They don't make the connection like that. So they kind of think that it's either not true, it's some kind of propaganda, which is direct. Sometimes they found that this kind of thing is directed to them. It's potentially threatening their trade. But still, they weren't sort of too bothered about us doing this study, and they were quite curious. I think they were quite curious also to see. So clearly, they don't see this as too much of a threat, certainly at this point. Now, in the market sellers, it was quite stark difference. So hardly none of them had heard of zoonotic diseases. So I don't know if there's probably, this is kind of like my deduction now, not directly from the study necessarily, but I'm just wondering whether there's actually a kind of income status difference. So the hunters are a bit more wealthy, so they have TVs, they got radios, they hear this stuff. The market sellers maybe they don't, because there was really a big difference in the understanding and how much they knew about zoonotic diseases. But so far they were happy to give us blood samples and cooperate in this. Like I said, I think a lot is because the hunters already knew our socioeconomic team, and they've got a good rapport and a good relationship. We talk very openly. We talk very openly about things. They ask questions, they are curious, and we try and share information, but do it in a way that we try not to put our opinions.
It's agonizing.
Yes, because this is also, if we look at this from the scientific point of view, we can't say anything for sure yet also, right? I mean, the indications are there that this is a risky interface. There are viruses that can potentially jump, so it's kind of there, but we do have to be quite sensitive about this. And whilst this trade involves relatively small amount of people, it's not like cattle farming or something that is very widely spread across, so it's relatively small amount of people, but for them it's very important part of their income. So we do have to be quite respectful of how we approach it at this point, and how we go about it. Yeah.
You say that it's small, and that's why you have to be sensitive, but it's big enough to affect the bat population, and you were emphasizing how important those bats are.
Yeah. So
it poses a threat to the bat population.
Yeah. So certainly, sort of continent-wide. So it's quite hard to put exact numbers on this. So IUCN has given it this kind of declining status now, and there are indications that the population of this specific species is declining. So the warning signs are there, but it's still quite difficult to put exact numbers of how many and what's happening. But this is quite slowly reproducing species. So any big declines in the populations would be quite slow to recover. And really, this is just unregulated. I think in most of the countries, this is unregulated. In Africa, we see definitely that this is increasing in Congo. It's very lucrative for the people. The only investment you need to do is buy nets. and then the bats will come seasonally. So it's obviously a very easy way of getting going. Minimum investment. If we have programs that are focusing on alternative meat sources, protein sources, to kind of reduce other wild meat and other bush meat consumption, and there are things like poultry farming and things like this, and they all require prior investment. So it's kind of easy to understand why this is a very attractive way of doing something for as long as there are bats. Yeah.
Okay, but you're taking the virus approach and not the conservation approach when you're trying to educate people. It seems from your answer that you're focusing more on that.
Our work right now is that, but every time we have a meeting and every time we do a look at funds or do a funding request to look for something new, we keep thinking, how do we bring up this conservation angle into it strongly, right? Because that is also very much at the heart of our activity. So whether this is one way of starting to look at it. If there is a risk, could we start regulating this kind of trade, and that would affect the populations of bats. So in these particular studies, the emphasis is on the risk of zoonosis and the impact of that in this trade. But I think I would speak to a lot in our organization, certainly, that we're all very concerned about the conservation status and the decline in the populations. And we constantly try and see ways that we could incorporate more effective ways of impacting that trend. Yeah. It's hard.
And even talking about educating people about viruses is a very difficult thing, because even highly educated people did not understand the whole virus part during the corona time. So yeah, that is a challenge.
Absolutely. I mean, we've been bouncing a lot of ideas, including with Sella, of how do we inspire conservation, and it's very basic things as well. And education is one of those things. We kept coming back to this kind of idea that you conserve, you protect what you care about, what you love. So we need to sort of start kind of also it's a long process. It's a long process, but that's kind of where it has to start and where it has to go, right?
Awesome. Thank you very much.
Thank you so much. That was a good question. Yeah. And I can see Karen, I think, wants to comment on this again. Before we go to Kirsten, Karen, you can make your comment.
Yeah, I just wanted to comment quickly on Iselda's question and comment there. I think when we talk about issues like this and how best to get the message across to these communities, we have to, from our side, generally Western conservationists that have had all our basic needs met, talking to us about the conservation of species, that makes a lot of sense, and that will move us towards action. But talking to communities that have very little, this is literally their bread and butter, they are far less interested in the conservation of some poor animal in the jungles, and a lot more interested in, can I make the money for my families, and can I keep my families safe and healthy? And so I think the approach of the health aspect, the potential of disease and harming your family is going to have a lot. We'll reach that conservation goal through the health aspect a lot quicker than, guys, stop this because we're trying to protect some bats. And I think that's
exactly why I asked the question, because in your previous talk about the snaring, you also mentioned the fact that the guy who did the snaring thought he was a hero providing for his family, because that is providing for your family, and that's how you were taught. The coolest guy was the guy with the coolest snaring techniques and whatever. So that was your purpose in life. And and that's exactly why I asked. The conservation part is so much more difficult to educate people if their basic needs are not met, like you said.
Yeah, I think it's a common issue, particularly in the global South. Finding a way to conserve species, by not talking about the species.
Yes.
It's one of the ways we got to do it.
Yes. Yeah. And like Sela said, like if you teach people to love something, then they will automatically conserve it.
Yeah. Yeah.
Cool.
That was a good discussion. Yeah. Okay. So I can see my colleague Kristen has been raising her hand for so long. Kristen, can you go for it?
Yeah, thanks, Stella. Thanks, Eva. Eva, sorry. Eva, a little bit on a tangent, a couple of questions that I have. The one is, what makes bats such, or carriers of such an abundance of viruses? I don't know whether I missed something, but it just, I don't know whether it's because they live in such large colonies or what is it?
Bats, this is a very hotly debated issue, and I'm like almost scared to answer this in much detail. But basically, bats are very special. So there's something about their immune system which basically allows them to have these viruses and not get sick. There are some sort of molecules in their system that, for example, dampen down inflammation. When you talk about something like Ebola, for example, a lot of the damage and a lot of the contributing factors that actually lead to a death of an individual is all that kind of massive destruction that your immune system does whilst that virus is going through your body. And bats' immune system is something quite special, and I'm not going to be able to tell you very kind of good explanation of how, but let's put it this way: it's not same as our immune system, and it deals with viruses very different way, which means that they might get infected and they raise antibodies, so the viruses are clearly recognized in the system, but they seem to just sort of eliminate the virus. The virus just kind of goes away without them mounting this massive disease response that happens in the rest of the mammals. And this seems to be kind of like what is happening with many diseases in bats. So they just kind of, yeah, I think they're sort of like a bit of super beings in this way. Yeah.
I just, my colleague Johan, I know he's hiding in the background, but he just said instead of bread and butter, bat and butter. So I thought that was quite funny.
Yeah.
Anyway,
Yeah,
my other question, was just around, are conditions, would you say that the conditions are favorable for Ebola to appear in other regions, in new continents, new regions?
Are the conditions favorable?
Favorable for Ebola to appear in new regions?
In new regions. Well, I mean, one of the things with Ebola, which is kind of like a very tickling thing, but we don't fully understand the kind of virus ecology of it yet. So where does it exactly lurk? Everything is pointing out to bats right now, but no one's been able to isolate, for example, Ebola Zaire virus, which is the causative Ebola virus in all these big outbreaks. No one's been able to actually isolate virus from a bat. So we kind of, maybe that's just the timing. It's very difficult to get it in that time frame, but we do see some of the antibody responses in the bats, but that is also quite low. So there's potentially still a bit of sort of missing links somewhere of understanding where does the virus lurk in between these outbreaks that we do detect. So where is it right now? We don't have an active Ebola outbreak in the African continent right now. So until we understand that, we can't really answer your question either very well. So if it's something like bats, I mean, when the 2014 outbreak happened in West Africa, no one was expecting it to go all the way that far. It was kind of, at that point, it was still all very much in the Central African region. So these things are happening, and if bats are spreading it, if these viruses are moving into bats, then we should be looking at their range of migrations. Yeah, it's possible.
Thanks,
but we don't know. We don't know much.
Lots of unknowns. So thank you.
Thank you so much. You can go ahead, Ken.
Sorry about that. Yeah, I got one more question for you. Your surveillance program your surveillance research that you were doing, where you spoke about Ebola, were you looking at any other virus or, well, any other pathogens aside from Ebola? And with that, did you ask the communities to report back any dead animals, or was it specifically great apes?
Yes, good questions. And this is kind of like a program also in development. So when we first started, it was very focused because it was kind of in response to these outbreaks happening. So it was very clear we were looking at Ebola virus and diagnosing only looking for Ebola virus in those samples. It wasn't solely great apes. Great apes was the sort of main focus because of this conservation angle initially. But we also know that other mammals do succumb to Ebola virus disease. So big mammals were kind of the target, but especially there has always been a big emphasis on great apes specifically because of the conservation issue. We've started to look now that our diagnostic capacity has also increased in Congo. So we really started from quite ground zero on that matter. The very first samples that we collected, we had to send out of the country, and then now we're at the point where we can do some of these analyses right next to the carcass with this kind of little portable PCR machine. But we've started to look at anthrax in these ones. Mpox has been a recent interest, so we're now looking at whether we could just retrospectively look at that just out of interest, and I think that is quite a possible target in the future, kind of on an ongoing surveillance basis as well. But then our kind of diagnostic capacity is increasing hugely, and these sequencing methodologies that are now emerging that are quite easy for us to introduce in Congo as well, they would really open up a completely different way of looking at. So we could actually take that same sample where we normally would just look at Ebola virus and actually sequence it all and look at hundreds of viruses in one go. So I think this is kind of in development.
Thank you so much. That was a good one also. And we also have a hand up from our director, Johan. Johan, can you go for it, please?
Eeva, thank you so much for 2 nights of, I would say, entertainment and education. So a question that I don't ask lightly, because I've been on the wrong side of the virus, quite significantly. But is a virus always a sign of a ecosystem that is out of sync, that is in trouble or unbalanced, when it's as prevalent as the case that you mentioned in the city with the bats? Or is there a natural role for a virus in an ecosystem?
Right. I'm not going to be the best person to answer this, and you'd probably get a really brilliant person for some of my colleagues who are really virologists and viral ecologists. But what I can say is that yes, there is a place for viruses in ecosystems. They are part of it. A lot of viruses, we all carry a lot of viruses. A lot of them don't cause any problems, but are integral part of how we also function. So there is this kind of ideal virus. We've been thinking about this a lot when we talk about these pandemics that are threatening and have been happening and all of this. And for virus, it's not ideal to kill your host, right? They need to have a live host. So it's kind of the problem is when these viruses that are in some kind of balance in an ecosystem get into a host that is not used to them. Put it that way, right? So when we talk about increasing risk of these spillover events, and we always talk about the balance of ecosystems and how intact ecosystems are protective in terms of public health as well, right? So as we go and disturb that balance that exists, that's where we think that the risk of these spillovers are increasing. So there is this balance, they are there, and And the more that we would have healthy, intact ecosystems, the theory at the moment is that those are protective of health overall and human health as well. Yeah. And when we see like I didn't go that deep into that difference that we see in the prevalence of the coronaviruses in Brazzaville and in Wesso, and coronavirus especially, so in those two towns that we've got in the north and in the south. And those two are very different kind of habitats now. And it's very kind of human altered landscape, especially in Brazzaville. So kind of hypothesis is a little bit that we would see higher prevalence of viruses in that kind of environment where there are much more stressors for the bats. Idea being, as we know from ourselves, we might be infected with a virus. Do we actually get sick, or is our immune system able to sort of eliminate that without us getting sick? Is connected to how stressed, how well are we in ourselves otherwise? Do we have good resources? Are we well fed? All of these same things apply to bats as well, right? And whether disease actually manifests in that equation of virus being present in there is a complex mixture of factors that are affecting. So that kind of environment, health of the environment and the area where these animals are living in, as well as us. It's very, yeah, disease ecology is complex, interesting, very fascinating area. And to try and understand these, there could be seasonal variations, the climatic variations, population density of the bats, and all these kind of things that are happening around them. It's like...
Thank you so much. These questions, they're very interesting, and we're getting interesting answers here. And I'm loving it. Yeah. To our audience, do we have any comment, any question before we close up for today? Okay. So as our director Johannes said, it has been 2 days of entertainment and education. We can just call it education, maybe. Yeah, thank you so much, Dr. Eve. We really appreciate your time, your commitment to this, and yeah, for sharing us this knowledge. Appreciate. And to our students, thank you so much for joining, for joining us for these hours. And also Karen, thank you so much. And the other name, I saw this name. Thank you so much. You've made this discussion very nice, and you've spiced it up. And good night to all of you.
Thanks very much. It's been a pleasure.